So welcome everyone. I'm really pleased to introduce David Engelthaler, who is, you were born and raised in Arizona?
Born and raised.
Born and raised. Undergrad at NAU, master's at Colorado State, PhD at NAU. Now director of TGen North, also the Health Observatory at ASU. He sort of splits himself into different places and time. We're getting sorted. And at any rate, we are very pleased to have him here today to talk about some of his work. So welcome.
Great, thank you, Ann. Well, thank you for the invitation. I'm thrilled to be here. What a cool group, and you guys get to talk about some great science. I looked at some of the past lectures. I'm going to hopefully discuss something that you guys will enjoy half as much as I do, and this is a lot of the work that I've been able to do in pathogenic fungi, Cocci and Crypto, so you'll see coccidioides, valley fever, and cryptococcus, which also we find in a lot of very interesting ways. And one of the big things that I've really been focusing on really is more thinking about paleoepidemiology from using genomics and understanding how these fungi have moved around the world and how they take on new phenotypes as well. And so we're gonna dive into it. Just so you guys, my background has been primarily in epidemiology, population health, genomics research, infectious disease for the past 30 plus years. When hantavirus first showed up in the Four Corners part of Arizona, which is in 1993, so well before anybody's memory, that's when I jumped into public health, and I was actually doing ecology and wildlife biology at the time, and that's why I really love pathogen ecology, epidemiology, and thinking about whole systems and how these systems evolve together. So I'd always like to start thinking about where we come from and understanding that these really are, when we talk about infectious diseases and pathogens, and the hosts and reservoirs, we really do have to think about the whole system, and the whole system is Earth. But we also have to think about where do we all come from, and we'll just do a little bit step back in time to deep time, just a couple 100 million years ago, thinking as Pangea is really starting to break up. We're actually starting to see that, while the majority of the landmass, pretty much all the landmass, is on one side of the planet, it has very different habitats and niches and climate, and overall the ecology is changing as the tectonic plates are moving. Just to point out, where by reference, there we are in North America, which actually looks like Africa, but it's actually North America. And then we fast forward about 100 million years. We're really starting to see the shifting of the plates and the formation of our continents. We can start to visualize them. And there's a point to all of this. And then about 66 million years, so about 40 million years later, certainly the continents are recognizable, but it's also, this is about sometime in this million years is when we had the worst day in the history of the planet, where an asteroid came in, landed somewhere where we called Chicxulub Crater now, in North America, or in the Central America, but that radically changed the environment, and it radically set us up for having new species form and species disappear. Obviously, all of our large dinosaurs disappeared, many of them on that day, but certainly in the ensuing months, they really all started to get wiped out. But it also set us up for a unique fungal ecology, and I'll talk about that in a minute, but let's get started on our two fungi. So coccidioides, my valley fever dream. So cocci is, hopefully everybody knows coccidioides, the fungal etiology of valley fever. Valley fever is it's really Arizona's disease, but it's found in Arizona and in California. This is a complete misnomer. I'm misbranding it by putting up this picture of the haboob, but everybody uses haboobs when talking about valley fever. Haboobs really have nothing to do with valley fever. We've been able to show this with new science and air monitoring. They really don't drive valley fever at all, but it's a great image to show. But just a little bit about valley fever to get everybody up to speed. If you're not, valley fever is, as I mentioned, it lives in the soils of the Southwest, but it really has two forms. One form is really a mold form that grows in the soils and can survive in the soils all on its own in this mold form, but really shows up in the thermic desert soils of the American Southwest. That's where it survives and thrives. But the spores from that form in the mycelia can break off if the soil's disturbed, gets up in the air, can be breathed in by a mammal, and then it turns into a parasitic form. And then once in the parasitic form, it's typically showing up as an infection in the lungs and can grow and replicate with different spore form over and over. It can actually end up either surviving long-term in the reservoir. It can be wiped out by the immune system, or it can be essentially walled off until the animal dies. And when the animal dies, the fungus essentially comes out of its form because the immune system's not holding it in check, and then actually starts to essentially digest proteins in the mammal and actually survives on the decaying remnants of the animal, and then goes back into the soil form of the fungus. So that's kind of this is really important to know, because we need to really understand the biology of this to understand how does it get out of the soil, how does it move around, expose, infect people, but also be dispersed around the world. I mentioned California and Arizona, so these are the two hot spots for this fungus. By and large, 90% of cases occurring, 90+% of cases in the US are occurring in California and Arizona, more cases in Arizona. But we also see it throughout the desert Southwest, New Mexico, parts of Texas, some parts of Nevada and Utah as well. But you can see that there's some spots across the country. That's just travelers. That's people coming to the Southwest, getting infected. Then they're detected once they go back home. We don't really see it's not that widespread. It's primarily just the desert Southwest. But also, you can see on this Western Hemisphere map, we see it down into Mexico. We see it in parts of Central and South America as well. So, again, dispersal is very interesting to me, and we got to think about what drives dispersal. We thought we knew. A lot of discussion, wind. We know that the spores can get in the air and wind can carry it, but how far does it carry it? And does that disperse it to new areas? Can it fall out and then land someplace and then start growing in? I don't think the biology tells us that. I think it's got to get under the top layers of the soil for it to really survive and thrive in new areas. Haboobs, I've already mentioned, this is not really an important driver of it. If the fungus is already there in the environment on the top of the soil, because construction or something, these big windstorms will move it around, but it's not a primary driver. Natural disasters, we've had some outbreaks occurring after natural disasters because so much soil is disrupted, say from an earthquake, like the Northridge earthquake, 1984, I can't remember when that was. 80s, and caused large outbreak, but did it cause it to disperse to new areas? We actually don't have any evidence that it did. Animals, we know animals are excellent vectors. They can move pathogens around and over long distances, especially large mammals. And then humans are the supreme vector in moving things around. We do that globally around the world all the time. And then climate change. Climate change is brought up oftentimes as a cause for this to show up maybe in new areas because the changing landscape. And in fact, somebody, a group out of Los Alamos, Morgan Gores, developed a model that if climate change was really driving it, then we would see, valley fever spreading really across the Western Hemisphere. Unfortunately, that doesn't really take into account the biology of the organism. So this is really more modeling fantasy than reality. We don't think that that's what's going to happen. But it's interesting to think about our changing environment. What is that going to mean for the fungus moving around to different areas? Just throwing this up here as an area of study that my team has been working on at TGen and now at ASU. But also working with CDC and the state health department and the local health department, Homeland Security, U.S. Weather Service. We are trying to understand this fungus where people are getting exposed in the air. For the very first time we've been doing this, we're looking at air filters across Maricopa County that are collected daily and tested. And we can start to relate it to weather factors. look at the correlations with dust storms. We were able to show there's zero, absolutely zero correlation with dust storms and haboobs, but then see that their landscape changes are probably a really important driver, which we knew construction probably pulls it up out of the soil. That makes sense, but we can actually, using some remote sensing and satellite imaging, we can actually start to do some predictive modeling as to where construction's occurring and potentially then where we're going to see at risk around neighborhoods and communities, depending on the size of construction over time. So that's a fun area that we're working on. This is really thinking more about cocci and exposure to cocci, not as it disperses around the globe. But so let's talk again about hemispheric dispersal. This was a great study done by a couple of folks that I've actually published with since then, Matt Fisher and John Taylor out of Berkeley, who were using some earlier genomic techniques. They were using microsatellites to understand the population genomics, population genetics at the time of this fungus. And they were able to show for the first time that the fungus actually has two species, what they were calling the California species and the non-California species. Of course, they're from Berkeley. That's how they labeled them. But it turns out the California species is what you find in California, and it is a species called Coccidioides immitis. Coccidioides possidacei is what we find pretty much in Arizona and then throughout the rest of the Western Hemisphere where it shows up. And they were using this, they were able to see that there were some phylogeographic patterns that where you were geographically, you would have distinct populations, meaning that you probably weren't having a whole lot of mixing, you weren't having continual dispersal. You had individual dispersal events that allowed for new populations to form and then be distinct from other populations. So this was great work. In this, they're trying to figure out how on earth it got to different parts, and they essentially thought, based off of their timing with microsatellites, which was fairly rudimentary but great at the time, that it was about 9,000 to 140,000 years old in South America, that something had driven it. And they thought, well, if it's 9,000, that means that maybe humans took it down into South America. Like we knew that this was a human disease. We weren't really thinking about this as a long-term environmental pathogen affecting the mammals into deep history. So once in the age of genomics, my team we did a project where we did pretty much the largest sequencing effort for valley fever at the time, where we sequenced a number of genomes and then gathered up everything else and developed kind of a new population structure understanding. Clearly we have those two species again, as I pointed out, but then we also used some tools, some Bayesian analysis tools, one called BEAST, to help us put a molecular clock, some timing on when dispersal or divergence may have occurred, and then time that to the dispersal to try to understand, okay, what may have been driving this. And so what we found is that the original population occurring in southern Arizona and northern Mexico was, or at least the first divergent population was occurring about 800,000 years ago after the species split many million years before that. Then we saw a population showing up sometime later in central Arizona and another distinct population coming out of the southern Arizona group showing up in Texas and in Mexico. And then out of that, we had the South American population showing up probably 550,000 years ago. And then another distinct population that was been found in Guatemala, only in one spot in a very arid location in Guatemala, came out of the southern Arizona population probably 150,000 or so years ago. And these are all fairly rough numbers, but they're giving us kind of an estimate, like what's older, what's newer, and maybe what had been happening about that time to help with this type of divergence. So let's go back to our map. Now we're 65 million years ago. This is, after the really the full breakup of the continents and after the Chicxulub explosion from the asteroid. And what we're starting to see is looking at North and South America. These are two disconnected continents. How would a fungus get from one continent to another? We go 50 million years ago, we're starting to get the shifting of the plates and uprising of the Panamanian Isthmus, but there's still vast distances between these continents. 20 million years ago, still not connected. and then, oh yeah, 10 million years ago. We're getting really close now. There's still some fairly big gaps, and there probably was some animal movement, certainly bird movement between the continents. And about 6 million years ago, when we see these series of kind of islands from the pushing up of this of these two plates, there were mammals like ground sloths that actually could swim and could swim from island to island and were able to move between the continents. But by and large, we were still blocked from having mass migration of animals. And that's what it looked like about 1 million years ago. But let's actually go back to about just before 3 million years ago. This is when we started to see the Great American Biotic Interchange. This is when we started seeing mass migration of animals, populations from both continents, finally seeing each other and in different continents. And we had some get wiped out because of carnivores wiping out new species. We had new species showing up, like our camels in North America shows up down in South America, which led to the llamas and alpacas and similar animals, and then in a wide variety of new species. But these species were carrying along with them the pathogens that they had. And so this is, it makes sense that they would have carried some things down from North America to South America. And in fact, Fisher and Taylor brought this up in that same paper from about 20 years ago, or 25 years ago, that this would have been interesting, but they're saying that would have been too long ago for this to have been a major factor, because they're thinking about, 2.5 to 3.5 million years ago didn't match up even with, it didn't match up with the timeline that they were seeing, which is like 140,000 at most. Well, we think this is probably closer to 1 million years ago, 800,000 years ago, at the earliest of the fungus moving between. So let's actually look at that on the timing. There were actually multiple of these mass waves of animal movements. They call the Gaby movements. There were 4 big waves of them, and really the third wave occurred about 800,000 years ago. And then the 4th wave occurred about 125 to 150,000 years ago. And we see that the timing of the fungus moving down certainly could have fit within these waves. We don't know what species may have taken them down, but we did have distinct species that are really affected and can carry this fungus long distance, like the canids. The Canis genus really started to move down right about this time, and that's about the time that we saw new populations showing up in Central America as well. So it's pretty interesting to think about the timing and the movement of animals for getting it to South America. Now let me talk a little bit about our C. immitis, Coccidioides immitis, which is what we see in California, which is primarily in the central basin of California. In fact, it's the San Joaquin Valley. That's where valley fever comes from. It's San Joaquin Valley fever. And that population is much newer, probably about 375,000 years ago did we start to see it diversify. So why would it take so long to diversify? And then another population for Southern California and Baja, Mexico showing up sometime later. And then this one tiny little population we're going to talk about up in Washington too. But we had to wait for the draining of the Central California basin. So what that is thinking about the Central California, where San Joaquin Valley is. If we go back 15 million years ago, before the continents were connected and before the subduction and pushing up of the Central or the California coast, there was no San Joaquin Valley. That was essentially the coastline of California. 3 million years ago, once we finally did have the connection, the Panamanian Isthmus brought up, that was about the time that we really saw a big giant inland sea of what's now the San Joaquin Valley. Sorry for the blurriness on this, but this sea would keep filling up and emptying out and filling up and emptying out with seawater for a couple million years. And then it would, once you had the full kind of completion of the coast up by Monterey, you actually it would drain out, and then it would fill up with fresh water from the melt from the Sierra Nevada glaciers would melt and then fill that up. And so the same kind of thing happening for hundreds of thousands of years, and then finally drained out about 700,000 years ago ish, and became probably the most fertile growing land, certainly in the country or in North America, but even in the world. And it has a lot to do with the salinity and other reasons for the soil. But it also is a very It's a great place. The high salinity and the very warm, arid conditions is a great place for valley fever and for or coccidioides fungus to grow. So that's why it took a while probably for that population to show up. But I did mention this Washington population. This one is of extreme interest to me because it happened, we just detected this not too long ago. And the question was, how did it get up into Washington? And is this something that's really more anthropogenic? And I think the answer is yes, but not in the way that a lot of people think. So in 2010, there were a couple of cases that showed up in south central Washington near the Columbia River, and we were able to identify what locations they were likely exposed to, and we're able to find positive fungus in the soil. And it's a pretty dry, arid part. It's not like wet Pacific Northwest. This is, again, south central. It's these drier regions of which then the fungus was found. We did the sequencing and genomic analysis on this to try to understand it. And this group of the Washington group seemed to be part of a San Joaquin Valley group and a much larger population that we were looking at, but it also looks very clonal. We called it the Washington clone. They're looking nearly identical, especially when compared to the other strains, but there is a fair number of mutations occurring between strains within this group to suggest that there has been ongoing evolution occurring, and we really think that it's about somewhere between 900 and 9,000 years old. We don't really have good numbers. We need more strains coming out of that region, but this is certainly pre-Columbian. So if it's anthropogenic, it's not because of modern day warming. It's probably something else happened for it to show up there, and this is one of the areas that I was very interested in is how has this been moving around, say in maybe in pre-Columbian time before contact with the Eastern Hemisphere. So this is a soil habitat niche model that was developed by CDC and others, looking at what is the best soil regions for cocci to grow. And it really is showing up primarily here in the Southwest, the Central Valley of California, which kind of definitely matches up where we actually find this. But up in Washington, where we see those cases, these are the two instances we find it in the soil right along the Columbia River. There is some of that darker orange there that suggests that it could be good habitat, but you look, it's completely surrounded by green, just terrible habitat, not cocci country whatsoever. How on earth did it get there? Well, let's look at a couple of places where this fungus has shown up outside the endemic region. One of those is up in northeastern Utah. Up in northeastern Utah is actually a place called Dinosaur National Monument. Hopefully people have been there, it's wonderful. But in one location called Swelter Shelter, there was positive soil and positive cases identified. But just one location, not in larger northeastern Utah or in that region, just in that one location. And then in California, in Northern California, there's been a few investigations identifying both in the soil and in human cases this showing up in these very distinct places, which is really outside the normal endemic range. The one thing these all have in common is that these are all archaeological dig sites. So as I mentioned, that one spot in northeastern Utah, Swelter Shelter, is a site where human remains and other remains have been found going back thousands of years. And then the same is true for the other sites in Northern California, where these were archaeological dig sites where human remains had been identified and exposed, dug up, going back several thousand years, 8,000 to 9,000 years, or 7,000 to 8,000 years on these. And what would happen is that they would dig in the students and the archaeologists, there'd be such enormous attack rates, 80 to 90% of everybody working on that site would get valley fever, and some of them very severe valley fever because they're just getting exposed to this fungus for the very first time for thousands of years is getting exposed. So it's kind of like digging up the mummy, but the mummy's the pathogen itself. The pathogen is there alive. It's not just been, waiting to be awoken. It's been surviving in the soil, but not have been exposed above ground. So these are probably people who had valley fever were buried in these sites, but they probably got valley fever from other locations and carried them to these sites. So what does that mean for Washington? Well, one of the things I thought was very interesting about this Washington situation is these two locations, the only place in the Northwest where this has been found outside those Northern California locations, is right here, this town, this is the town of Kennewick. Kennewick is famous really for one thing. Kennewick Man, the ancient one. Right? When the remains of this individual were found, this was the oldest known complete human remains found in the Western Hemisphere. Think about 9,000 years old, considered. I think that was what it was estimated. Of course, now remains have been found for much earlier than that, going back 13,000, 14,000 years. But at the time, it was the earliest. Now, it's not to suggest that Kennewick Man, or the ancient one, was infected with valley fever. But what has since been well understood is that there have been multiple eons of people being buried along the Columbia River as populations have grown and lived there. So it's very possible, and what we think is very likely, is that an early individual infected in Central California, moving up through trade, or maybe a mammal, like a dog, being moved up and was then buried, because also canine skeletal remains have been identified along the Columbia River as well. And it couldn't have been obviously too much before that. Humans didn't come in too much before that, maybe again 15,000 years, but not in that region, because that region was completely flooded as the glaciers were melting. These Missoula floods lasted for thousands of years, and that was all underwater at the time. So we do have a sense that this is probably a little bit earlier, 9,000 years in that region, and then maybe, just maybe, the conditions have changed enough, maybe from more recent warming, that has allowed things to be really more revealed that were already there for thousands of years. In fact, that's probably what this slide says. Yes, this hypothesis, which then we've published on a few times. I actually did this study with Jim Chatters was part of this investigation. He's the one who did the first description of the ancient one back when it was found 20 plus years ago. All right, so that's pretty much what I was just saying. So all of that's to say that we've really thinking about both dispersal and exposure, but dispersal of this fungus, we have to think beyond people getting infected, which is what we do with pathogens all the time. We think this is a human thing. It's not a human thing. Humans are just accidentally infected, but humans actually can have a really important impact on dispersal. And then we think about weather, right, because of the dust storms. So there's been multitude of modeling studies and other investigations to just try to look at linking weather to human cases to develop a model for why valley fever occurs. But in reality, what we're identifying is it's a much more complex system, and we're really starting to understand then both microdispersal and macrodispersal, how that changes the population structures of these fungi, and the different kind of clues we can look for to see when and where and how this fungus moves around the world. Okay, we're halfway through, a little over halfway through. But that was our one fungus. Now I want to talk about Cryptococcus, the curious case of Cryptococcus. So Cryptococcus, another environmental fungus, typically had been seen in tropical and semi-tropical areas of the world. This fungus itself really survives and thrives off of decaying tree matter. So, where you have rainforests and whatnot, where you have a lot of decaying trees and shrubs, etc., this fungus thrives, especially in tree holes. And then if it's disturbed, if you can get essentially either yeast or spores up into the air, and you can breathe that in, and you can develop a pulmonary infection. Of course, humans, again, very accidental, actually not even important, because this one, if it kills a mammal, it typically doesn't go into digesting the proteins of the mammal to survive to get back into the soil. It's pretty much a dead-end host. This thing is really just surviving and thriving in the woods, rainforests, etc., and then mammals can get infected as dead-end hosts. What we see, again, primarily a pulmonary disease, but it can get out of the lungs and get into the CNS and cause meningitis and encephalitis. And the sister of the Cryptococcus, so we're talking about Cryptococcus gattii, the sister of that, Cryptococcus neoformans, is actually the #2 killer of patients with HIV in the world, right behind tuberculosis. And so it really attacks the immune incompetent and has a high fatality rate that way. What happened Oh, and I'll mention that it's been found throughout the tropics, so it does infect other mammal species, and you can see these are both the two patients, including this. Okay, I don't need help, thank you. Try and tell me about warm tropical climates. We did a study with koalas and understanding the populations that were moving in koalas, and we saw that some koalas moved from Western Australia over to Eastern Australia, actually carried the fungus and started a whole new population. So it was koala transmitted, but anthropogenic because we were moving the koalas. And in fact, I was able, down in Australia, I only show this picture. That's me pointing at a koala. My colleague from UC Davis took this picture, and right after this, that koala peed on me. Did not spread crypto as far as I know, but just kind of a funny picture. But really what was of interest is this fungus all of a sudden shows up Pacific Northwest. Again, the Pacific Northwest, why do we have a fungus that doesn't belong there? It's a completely different niche, different part of the world, and all of a sudden starts causing a lot of cases. It emerged on Vancouver Island right around the year 2000, 2001, I think the earliest case then we were finding back to 1999. And then the more they looked, of course, the more they find. That's what happens with disease surveillance once you finally know to look for something. And they found quite a few cases occurring on Vancouver Island. So what were we seeing? What we were seeing is that it was infecting really immunocompetent people. We were seeing a pretty high fatality rate in immune competence. So this wasn't kind of following the pattern of people that have different kind of immunodeficiencies. And then we were seeing essentially a new strain. Forgive the nomenclature here, VG2A. VG2 is a major molecular type. It's probably its own species of Cryptococcus that fits within kind of the larger Gaddii species complex. So this kind of new strain that we didn't really know of anywhere else in the world shows up in around 99. And then there was an outbreak in Oregon and Washington a few years later, but that's primarily because we were looking for it. It was probably all occurring pretty close to the same time. And this was also, in Washington, we were starting to see another strain, very closely related, but very also distinct. And these new genotypes, again, I was mentioning, have new phenotypes. So you're in a different niche. This is a temperate niche. It's not a tropical or subtropical niche, but it is very rainforesty, if anybody's been to the region, which means there's a lot of decaying wood matter for the fungus to feast on. And we were seeing primarily just staying in the lungs, as opposed to going to the CNS, which sounds good that it wasn't getting up into the brain and the meninges, but it was actually causing a much higher fatality rate, about 25% fatality rate. Probably wasn't that high because it was the first cases that you find are always the most severe, but still a really high fatality rate. So it's multiple new phenotypes happening with these new strains showing up in a region that we definitely didn't expect it to show up. And this is showing kind of the location where cases were. Willamette Valley, which comes off of the Willamette River, Columbia River, and then up in Puget Sound. Okay, so I mentioned these multiple new strain types. Initially, CDC and other researchers, they used MLST to look at the diversity within these, and they all looked very clonal in these new sequence types. So not very many differences. So CDC came to us and asked, can we do some whole genome sequencing, because there wasn't a lot going on in fungus at the time, to see if we can understand what really is happening there. And we found surprising amount of diversity within these groups. So these species that were showing up there, these populations, were not brand new. There was definitely some local evolution occurring in these strains that are really only found in that part of the world. At least both the VG2A and VG2C. VG2B is a little weird and shows up in multiple places around the world. So what did this mean that we're seeing these new genotypes and trying to understand why was it causing new phenotypes? We did a fair amount of the classic molecular genomic studies. We did whole genome, looking at gene content and differences between the populations, doing transcriptome analysis, growing under different conditions, trying to elucidate what were the genomic features that were causing these phenotypes. We did find a few genes that were existing in some species up there in the Pacific Northwest, but not in other crypto species. So we had some clues. We're starting to get in doing some CRISPR analysis to really start to plug and play on some of these genes, which is not gain of function. But then the pandemic happened, and we kind of just shut all that down. But there's multiple other stories behind this. I'm not going to really go into this piece, but what I did want to do is understand, again, how did this fungus get here? When did it get here? Why did it get here? And so that we can better understand, maybe will it be showing up in other places? So by doing the analysis on this, we did find a couple VG2As in other parts of the world. They're essentially only in Brazil, and it seemed to have been found near a port city of Recife, Brazil. So that seems like, okay, maybe that's where it came from, thinking about Recife. So when did it get there, and then how did it get from that port city, if that's in fact where it came from, up to the Pacific Northwest? Well, first we had to see how old this was, and again, using beast analysis and much more precise beast analysis, we had lots of strains to work with. We were really seeing that these strains really are about 70 to 90 years old, at least when they really first started diversifying was about 70 to 90 years old from the time that this analysis had happened. And if it really did come from Recife and go up to the Pacific Northwest, how did that happen? How did that happen? That would have been probably then now 100 years ago, moved from that side of Brazil up to North America, because there really was no direct trade routes. Sailing ships would essentially have to come all the way around the Horn of South America, all the way up. And we're trying to understand, was that a feasible way for material, something to allow the fungus to be transferred up there? Well, we came up then with the Teddy Roosevelt hypothesis. The timing on this was somewhat suspicious. 110 years ago, this is when we carved the Panama Canal, and now ships can actually sail through the Panama Canal carrying a lot more material from the eastern side of South America to the western side of North America. And then we had a lot more shipping happening as we're getting a lot of growth in Vancouver and the Puget Sound, Seattle area as well. So that actually made sense timing-wise. Probably had to wait till this kind of shipping lanes were open. But then I try to figure out what would have been transferred. How on earth do you infect a whole region by bringing material from one part of the world to another? And one of the things that we know is, and I looked at a lot of shipping manifests, really cool, 100-year-old shipping manifests. Did we move rubber tree plants or lumber or whatnot? We really couldn't find a good suspicious target. But all ships, even today, especially then, would take up ballast water, and then as part of, to balance their load as they're going through the ocean, and then they would dump ballast water once they get to the new harbor. This is really common practice, and when you do that, it's not just water, it is everything that's in that water, including microorganisms, including even small fish and shellfish and others get transported around the world that way. But we do know that the fungus could have, because this fungus is shown to survive in the water. This Cryptococcus survive in seawater, and really importantly, what we were seeing is that there were a lot of marine mammals, once we started looking for this, actually were infected with crypto. We were identifying porpoises and dolphins, seals, sea lions, whales, a number of different species that were being infected in all of that Puget Sound area, the Salish Sea, and forget else what they call it, but around Vancouver Island and down even into near Seattle region. So the fungus is definitely in the water, and it's definitely infecting animals. But the question was, did it start in the water? And if it started in the water because it was transported in ballast water, how on earth did it get up on land, causing all these other infections? So these little red dots and the little squares by them is all humans and their pet dogs, essentially, that were getting infected. But they were all getting infected along the coastline where these cases were. Very rarely did we see anything showing up internally. Occasionally we would, but certainly along the coastline. So one of the things we went back and looked genomically, it looked like there were two diversifying events. And was there potentially a second emergence occurring within these? And it looked like there was about 20 to 40 years later. And if we look on our history, one major event occurred, the Alaskan tsunami. So the Great Alaskan Earthquake, 9.2 on the Richter scale, is the largest earthquake in recorded North American history. Caused massive, then not just damage on land, but massive tsunamis and flooding for a very long distance, including all the way through the Puget Sound area. And then that is the one thing that would have pushed contaminated water tens of meters up on land, based off of the documentation. And certainly any microbes with that would have well seeded the forested region around the coastal areas where we see it, even within the Columbia River down into Willamette Valley. So it's one of those where it's pretty theoretical and hypothetical, but it is the only story so far that makes sense on how you can get a individual or a couple individual populations from one side of the hemisphere to the other side of the hemisphere, going back about 100 years. Okay, so I did mention something about dinosaurs and fungus, and I just wanted to touch on this real quick. One of my colleagues, Arturo Casadebal, brilliant, out of Harvard and Johns Hopkins, he actually had been proposing a theory about how the asteroid that wiped out the dinosaurs actually set up the globe for massive expansion of fungi and fungal species. And it really does make a lot of sense because, we did have this kind of almost like this nuclear winter effect where the sun was blocked out for months on end. It caused giant global cooling, which obviously aided in the getting rid of our ectotherms. Our dinosaurs were all wiped out, except for our birds. And then there was the rise of the endotherm. So then, because it was so cool, animals that could make their own body heat really did survive and thrive. But because it was cool, fungi also survived and thrived. It was a very warm environment before. You had limited pockets probably of fungus before this event occurred. But this then allowed for us to have a fungal Earth because of the impacts of the asteroid. Arturo Casadebal and John Taylor, as I mentioned him earlier, they came up with a theory also on how the separating land masses from Pangea actually started to separate species out. We know this happened for many different types of species. They proposed that this likely happened with fungi, including with crypto. And so I went back and mapped what we call the different crypto species now, part of the different species complexes, and understand where they are now as opposed to where the continents were before. And we did find some evidence that would make sense that the separation of the land masses would make sense. For instance, in India, you see this one species called VG4, really the only other place where we see that dominant. We've seen introductions in the Americas, but dominant is in Africa. And India used to be connected as a landmass to Africa. It's possible it happened later on, but these are really old populations, and so it is possible that these got separated by the separating land masses. So again, just some fun stuff. Oh, I skipped a slide. That's right. So there, that was some of the fun paleo epidemiological work I've been doing. Just a reflection, again, my time in Australia as the world keeps changing. Don't forget to stop and pet a roof from time to time. Epilogue, our world will indeed keep changing. The tectonic plates keep shifting, and that means that we're not in a static environment. Our environment, our climate, everything will continue to change over large periods of time. We start to see that the projections on the changing tectonic plates, that Africa might really join up with Europe, and then the continents might actually end up coming together. This is some projections, maybe 250 million years from now to have Pangea 2, and I can't wait to see what that looks like. All right, thank you. Questions?
Fantastic. Loved everything about your talk. And I have a lot of questions about how, I'm going back to the desert valley, coccidioides, or however you pronounce it, being mammal specific. And I was thinking, most birds aren't digging into dirt, but I immediately thought of burrowing owls and turkey vultures because their range exactly mimic. I mean, they're desert adapted, and their ranges mimic very clearly the distribution of where we're seeing desert valley fever. And so I'm curious as to why birds are so resistant to this. Why, and whether or not they could potentially spread it even if they don't become vulnerable to sickness from it, and how that works, if you can speak to that at all.
Yeah,
vultures are eating like decaying the mammal tissue, right? That the fungi are consuming before returning to the soil, and the burrowing owls are going quite deep and relative to any other birds in terms of that they're digging in soil and going down into it. So I'm immediately curious as to how mammals are the specific target, and what about birds makes them resistant to the extent we know, and could they be carriers even in that resistant state?
Yeah, that's a great question, and it's been thought about a lot as to why we really only see this as a mammal pathogen. Probably has something to do with the average body temperature differences and how the fungus has evolved to really survive in mammal level body temperatures. There are probably other components that allow for survival within the mammalian lung that may be different. Don't think we fully know as to why, but it really is, it's really seems to have co-evolved with mammals.
Are there fungi that are like are there fungi that are commensal with birds that would basically be a competitive inhibitor of the establishment of these guys, or do we know anything about fungal lines that are specific to these big taxonomic groups?
Yeah, probably not so much a competitor that would prevent them from being infected, but likely, so let's just think, with mammals, there are relatively few fungi that actually infect mammals. Fungi are really more cold adapted, and there's very few that have become adapted to infecting mammals, and the ones that do are usually cooler environments, usually on the skin, hair, and then the ones that actually get inside, this is just a very small number. So there's probably not a whole lot of competition that would be out there. The bird immune systems may have developed in a way that really allowed them to survive. Birds are the last surviving member of dinosaurs, right? And so that was happening at the time when at least theoretically, we have these massive fungal blooms. So they may have developed some very useful immune responses to prevent these types of fungal infections. And you mentioned birds. I think flying mammals, though, might be really important. So or I shouldn't say really important, might be important in that, so bats have been found positive for Cocci down in South America. It's possible that they can act in a way that's providing this long-distance dispersal. Maybe it's bats that took it up to Washington. We don't have any evidence of that. And if that was happening, it likely would have been happening multiple times, right? We have the ongoing migrations. We don't see that. We see one exact population showing up in Washington, just one event. And we see distinct populations in Central and South America. So that's most likely something that's occurred once rather than an ongoing type of migration.
Well, thank you so much for this nice talk. It's so reassuring to know that people are doing this type of research living in Arizona. Well, I have two questions. They're actually genome, a bit like technical, specific question. I've worked on fungus, and I know it was a nightmare to sequence the genomes of this fungus, of some other fungi, and then you actually, while people are doing their phylogenetic on MLSTs, you were doing it on the whole genome sequences. I actually wanted to get a bit of insight from you, like how you successfully manage all those things. And on top of that, I have a related question. So the fungi that you were talking that they're emerging, I believe they are the clinical outbursts. So like there's some new people detected, and the isolates you have are the clinical isolates. So is there a possibility to go and then look in the nature and then see some guys who are actually not doing the sicknesses, but they are there and they're kind of closely related, and then design a comparative genomics approach to see, while some of them are hanging out in the forest or in the soil and the others are causing the infections? I'd like to get some expert opinion on that.
Yeah. Oh, well, I'll give an opinion anyway. So expert opinion. So a couple of things. Yeah, the sequencing fungi versus, say, bacteria and viruses, much more complex. We had to really wait till we had these very kind of high throughput sequencers, very large space versus some of the earlier sequences. We actually did, my lab in Flagstaff, we did the first genomic epidemiology investigation of a fungal outbreak ever, which was a valley fever outbreak in people who received organs from the same donor, and they all developed valley fever. CDC came to us and said, can you do that genomics thing you guys do to see if these things are related? And it was a lock, they were related. They were all identical genomes. We were using some older technology before the Illumina instruments came out, but it cost a lot of money and it was a lot of horsepower to do it, but we just had to prove that we could do it. And now, ever since then, with Illumina technology, we can get a lot of sequencing depth, and it's just a matter of which tools that we need to use for for assembling, because in many cases we're doing this for the very first time, right, as opposed to just aligning other known genomes. But once you have some good assemblies, we can do the analysis. Excuse me?
How big are these genomes?
Oh, so we could be, anywhere from 10 million to 50 million bases. Yeah, so not human genomes. And so I actually brought in a human genomicist to help us with that very first fungal genome sequencing that we had done. So yeah, relatively speaking, they're very large in the microbial space, and they have a lot of complexity, and especially in population analysis, because they're doing in a lot of cases you have both sexual and asexual reproduction. You have picking up just random chunks of genomic material, not even fungal sometimes, and somehow incorporating that. So we get these very complex populations, and thinking about really focusing in on the core genome allows us to develop these populations better. So there's more we could talk about that. The other part, I think it's great, is that more and more people around the world are just doing environmental metagenomic sequencing, and and publishing. And NCBI actually has great databases of which people are putting all their metagenomic data, that you can go through and look for signal of different fungi showing up in different parts of the world in samples that you weren't actually looking for fungi. We've done that in looking in say, like, water that was sequenced by, pulled from Puget Sound, to find the actual presence of Cryptococcus in those water samples. So really relying on other people's science to help us identify signal of where maybe new fungi are emerging. And when we say emerging, it's only emerging in our consciousness. Sometimes this fungi are around there the whole time. We're just seeing it for the first time because we're looking for it. One last example, we had a really nasty outbreak of a fungal infection caused by a fungus called apophysomyces after the Joplin tornado. The Joplin tornado 14 years ago ripped through, just tore up the Joplin, Missouri really badly, infected 13, 14 people with a fungus that we'd never seen cause an outbreak before. We've heard of it because we've studied it in the soil before. Every once in a while you get these weird human cases, but this is the very first outbreak ever recorded of this fungus. And so then we could do some population analysis, understand that fungus was probably there for 1 million+ years in the soil and never causing an infection. It was only because of this tornado, the natural disaster, that people got exposed to it, lacerations and all that, really bad outcomes. But it wasn't an emerging infection. It was there all along. It was just we got introduced to it by a tornado. So there's a lot of different scenarios in which we're going to find fungi that are emerging. I don't even know if that answered your question. It's just a fun story.
So what you just brought up actually relates to my question as well, which is, how does sort of the reproductive cycle or the replication time of these different fungi impact your, estimates of dating, when they may have arrived or when they went, their most recent common ancestor with others. So like thinking about the Washington cases,
mm-hmm.
right? Or these other ones that are linked to sort of, human remains that have been underground for a really long time. Is there some like relative dormancy where maybe they're not replicating as much, and so the number of mutations that are coming up need to sort of recalibrate where in time, they their most recent common ancestor is?
Yeah, well, it's a great question because you really do have to understand the biology of these organisms in the type of environment that you're looking at. You saw that the actual rough estimate was 900 to 9,000 years. Because we really don't know what has been going on with that fungus. Has it been circulating and maybe even infecting local rodent populations, and which is going to, have a higher mutation rate most likely as it's cycling through animals and then getting back into the soil than if you just have it growing as a mold in maintaining that, which it could on its own as well. So trying to have both an upper and lower extreme in an estimation like that, it's a really important point that it goes back to us. We can't just do genomics. We can't just do fancy bioinformatics. We truly have to understand the system that we're talking about and incorporate those as priors into our analysis.
I think we're at the end of our time.
I think we are too.
But please join me in thanking Dave for coming and speaking.
Thank you, Ann.
Fascinating.
Yeah,
talk.
appreciate it.